Good afternoon, guys, and today we will continue the physiology of the urinary system. And in the previous lecture, I have discussed the various functions of the kidney, like metabolic, excretory, endocrine, and regulatory. So you can check the previous video. Today, the table of content is the renal anatomy, just the basics and the nephron structure, types of nephrons. The main thing is about the cardiac output and the importance of the minimal urine production. So let's get started. First of all, this schematic diagram is representing how the kidney is supplied by the renal artery. This is the abdominal aorta. Um, Projecting from the abdominal aorta is the renal artery, as you can see here, and as it progresses towards the kidney, it divides into the anterior and posterior divisions. The posterior division supplies the posterior aspect, and the anterior division, first of all, forms five segments, segmental arteries, as you can see here: one, two, three, four, and five. It forms five segmental arteries. From the segmental arteries arises the interlobar arteries. Okay, interlobar arteries are running in between the pyramids. You can see here, this is one pyramid, this is the second one, this is the third one, and they contain about 8 to 12 pyramids in each kidney. Sorry. So the interlobar arteries run between the pyramids, and then at the junction of cortex and medulla, this one, I hope you can see it. I will highlight it. This is the junction of the cortex and medulla. Here the interlobar arteries divides to form the arcuate arteries. Okay, arcuate arteries. As the name suggests that it forms the arc structure between the cortex and medulla. From the arcuate arteries, I will zoom it so you can see it more clearly. From the arcuate arteries arises the interlobular arteries. Okay, and finally the interlobular arteries progress to form the afferent arteriole, which forms the glomerular tuft of capillaries and exiting as the efferent arteriole, and after that they form the peritubular capillaries. So let's revise it. From the abdominal aorta arises the renal artery. Renal artery anterior division has segmental arteries. These are the five segmental arteries: one, two, three, 4, and 5. From the segmental arteries arises the interlobar arteries between the pyramids. At the corticomedullary junction, arcuate arteries are formed, and from the arcuate arteries, interlobular arteries, and finally the afferent arteriole forming the glomerular tuft of capillaries and exiting as the efferent arteriole. And this is the special arrangement in which the glomerulus is arranged between an arteriole and exiting also in between an arteriole. This system is high pressure system because normally the in body they are arterioles, then they are capillaries, then they exit as venules, but here is the special arrangement. This is an exception. Okay, and the efferent arterioles continue to form peritubular capillaries, which, as you can see here, these are the peritubular capillaries. They are around the proximal and distal convoluted tubules for the assisting the reabsorption process. So I hope you understand this diagram. So let's just uh, collect the things together. First of all is the cortex, is the outer layer. It contains glomeruli. As you can see here, these are the various glomeruli: Bowman's capsule, the proximal convoluted tubule, this one, and the distal convoluted tubule. The medulla. Medulla is the deep structure. It contains the collecting ducts, the pyramids, and the loop of Henle's. Renal calyces are the structures. This is the renal pyramid. So the apex of the renal pyramid is known as the calyces. Okay, these are the calyces. Okay. After that, the renal blood supply, as I've told you, from abdominal aorta, renal artery, interlobar, then arcuate at the junction of cortex and medulla, then interlobular, efferent, glomerular capillaries, efferent, and then peritubular capillaries. There is a difference between the kinds of nephrons in which they do not have the peritubular capillaries. So, okay, the blood network. As I've told you that glomerular capillaries are high pressure system in which there is an afferent arteriole and efferent arteriole. The peritubular capillaries surrounds the PCT and DCT for the reabsorption process. What are vasa recta? Vasa recta are the specialized descending loops which are present only in the juxtamedullary nephrons. What is the difference between the vasa recta and the peritubular capillaries? Because both are around the vessels, around the tubules. So let's see here. This nephron, which I am highlighting in blue, okay, and here's another nephron I'm highlighting in purple. This is known as the cortical nephron, and the deep to it is the medullary nephron or juxtamedullary nephron. The cortical nephron, in which the efferent arteriole forms the peritubular capillaries, but in the juxtamedullary nephrons, the efferent arterioles form the vasa recta. Okay, vasa recta in the juxtamedullary nephrons. So let's see here in more detail. Types of nephron. Collecting nephrons, they are present in the outer cortex and the juxtamedullary at the corticomedullary junction or deeper to the cortex or in the medulla. Cortical nephrons are more, about 90% of them are cortical, juxtamedullary are less. They have shorter loops of Henle and the juxtamedullary have the longer loops of Henle. In the cortical nephron, the main difference is that the efferent arteriole forms the peritubular capillaries, while in the juxtamedullary, the efferent arteriole forms the vasa recta. Here's a diagram. You can see here clearly that this is the cortical nephron and here is the juxtamedullary nephron. So it has the shorter loop of Henle and it has a very, very long loop of Henle. In this, the efferent arterioles form the peritubular capillaries, and in this structure, the efferent arteriole form the vasa recta for the. And what is the importance of vasa recta? Vasa recta has a little blood supply for preserving the osmotic gradient, and it assists in the reabsorption process. Okay, and in the formation of urine, they are very important. As you know that Bowman's capsule, it's a double wall structure, PCT, loop of Henle, DCT, and the collecting duct. The collecting duct is also known as the duct of Bellini. Okay, so this one was the basis of the renal anatomy and the blood supply and the classes of nephron. So let's discuss about the some urine formation and physiological aspect of it. The feature of glomerular filtration is that it is situated between two arterioles, so it is a high pressure system and it is supported by the mesangial cells. What is mesangium? Mesangium is this structure which is highlighted in dark blue color. It is mesangium. Okay, it is mesangium. Um, Mesangium, it is a connective tissue. Okay, a mesangium is a connective tissue and it is located between the glomerular capillaries, and the function of the function of mesangium is to support the glomerular capillaries. and it is a connective tissue. It is also containing the mesangial cells, which is important in the secretion of various hormones and substances. We'll discuss them later. Okay, so coming towards the cardiac output and the renal blood flow, it is not the blood that supply that we have discussed, but it is somewhat different. Let's discuss first of all the cardiac output. Cardiac output is defined as the volume of blood pumped by the left ventricle in 1 minute. What is the formula for the cardiac output? It is heart rate into the stroke volume. Heart rate, as you all know, that it is approximately 72 beats per minute. What is stroke volume? It is volume of blood pumped out of the left ventricle during each systolic cardiac contraction. It means that each time a heart beats in a systolic contraction, the amount of blood pumped out of the left ventricle, it is approximately 70 ml. So the formula was the heart rate 72 into stroke volume 70. It becomes approximately 5000 ml. The units are ml, and ml can be converted into liters. 5 liters per minute. So the cardiac output of the heart is 5 liter per minute, 5 liters per minute, and it is in each minute the heart is supplying the body 5 liters of the blood. So let's discuss the both theory and the diagram side by side. Okay, this is the heart, and the cardiac output is approximately 5 liters. From the 5 liters, the blood comes towards the general circulation, and this is the renal artery. This is renal artery projecting from the general circulation, and the renal artery carries up approximately 20% of that blood. 5 liter is produced. From the 5 liter, 20% goes to both of the kidneys. In this diagram, we are discussing both the kidneys, not only one. Okay, both kidneys are being discussed here. So, 20% of 5 liter is 1 liter. It is 1 liter. Okay, 1 liter can be written. There is no mistake in writing it as 1000 ml. Okay. From 1000 ml, it contains blood. So blood contains the cell and plasma. Plasma is 600 ml, and the cells like RBCs, WBCs, and platelets contain 400 ml of the 1000 ml. Cells are not filtered, they just pass through the glomerulus. They are not being filtered, but plasma should be filtered because plasma contain all the ions, water, and essential nutrients which we need to reabsorb and process in the urine formation. So let's revise. From 5 liter, 20% goes to the renal artery. It is 1 liter, so 1000 ml, and for 400 ml from 1000 are not filtered, they just pass through. 600 are in consideration. From 600, because the plasma contain proteins, plasma also contain proteins, so from the 600 ml, 20% is filtered. What is the 20% of 600? It is approximately 120 ml. 120 ml per minute. We are talking in 1 minute. Okay, but it is 120, so we can write it as 100 for easy understanding. Okay, 100 ml per minute of plasma is filtered in glomeruli of all nephrons in both kidneys. Okay, so 20% is filtered. Approximately 100 ml comes towards the glomerulus. So let's talk about each structure. In the proximal convoluted tubule, 65% of reabsorption occurs. 65% of 100 means that 65 ml. 65 ml is reabsorbed in proximal convoluted tubule. In the descending limb. In the descending limb, approximately 15% reabsorption occurs. So 15 ml is reabsorbed. So 80%, 80% is reabsorbed in the PCT and the descending limb. The descending limb. In the ascending limb, this is the ascending limb, there is no reabsorption occurring because it is impermeable. The walls of the ascending limb of loop of Henle or thick limb of loop of Henle is impermeable to like water and the solutes dissolved in it. So there is no reabsorption occurring in the ascending limb. So proximal convoluted 65%, descending limb 15%, so it becomes 80%. Coming towards the distal convoluted tubule here, up to the distal convoluted tubule, 80% of the plasma is reabsorbed. In the distal convoluted tubule, 15% is also reabsorbed. So here 95% is reabsorbed from the 100 ml, so it becomes 95 ml is reabsorbed in both kidneys in 1 minute. Okay, so what is remaining? It is approximately 5 ml. 5 ml is moving forward and the fine tuning occurs in the collecting ducts. Collecting ducts are influenced by the hormones like antidiuretic hormone, vasopressin and aldosterone. Aldosterone is produced by the adrenal cortex, and under the influence of both these hormones, the 5 ml is tuned and the water is reabsorbed depending on the need of the body or the metabolism occurring inside the body. 1 ml come as the urine output. It's very astonishing that from the all the from the plasma only 1 ml of the urine is produced from both kidneys, from both of the kidneys in 1 minute, both of the kidneys in 1 minute. Okay, 1 ml of urine is produced by both kidneys per minute. So let's revise it so you can gather the things together. 5 liter was produced by the heart, the blood. 20% goes to the renal artery. 1 liter, it can be written as 1000 ml. From 1400 ml are cells. They have no function in the urine formation. 600 ml is plasma. 600 ml also contain proteins like albumins, globulins. They are not being filtered. Only 20% of plasma is being filtered. 20% becomes 120 ml. But for practical purposes, we can write it as 100 ml. 100 ml is moving forward in PCT, 65% is reabsorbed. So in the descending limb, 15% is reabsorbed. So 80% is reabsorbed in this area. Ascending limb is impermeable. Distal collecting tubule offers 15% reabsorption. Up to here 95% is reabsorbed. In the collecting duct, fine tuning of the urine occurs, and under the influence of hormones, the 5 ml is also reabsorbed and only 1 ml urine is produced in both kidneys in 1 minute. You know that both kidneys consist of 2.6 million nephrons. So this is the representation of only one nephron, but you can imagine that this process is occurring in all of the 2.6 million nephrons and they collectively produce 1 ml of urine 1 minute. Okay, so 1 hour contains 60 minutes and 1 day consists of 24 hours. So 1 day can be written as 24 multiplied by 60 minutes. It becomes 1440 minutes. So we discussed that in each minute 1 ml is being produced. So in 1440 minutes, 1440 ml Approximately 1.5 liter of urine is being produced daily. 1500 ml produced daily. It is like an average amount of the urine produced, but it can be varied in different persons physiologically as well as pathologically. So let's discuss what is the normal range of the urine. Normal output of the urine is in between 500 ml to approximately 3500 ml. Why is this range dedicated as 500 and 3500? Let's first talk about the maximum urine output. It's 3.5 liter. Exceeding this amount will produce nocturia and disturbance in the sleep. Nocturia means that the person has to wake up during the night and he has to empty his urinary bladder approximately 5 to 6 times a night. So it's a very difficult for a person to maintain the healthy lifestyle. Quality of life is disturbed. And what is the importance of minimum urine output? So let's discuss it. Why 500? Why not 400? Why not 300? Let's discuss. Kidney concentrate the metabolic waste like urea, creatinine, and other salts, bile, etc. This is concentrated by the kidneys. The maximum concentration capacity of the kidney is 1200 millimoles of metabolic waste in 1000 ml of urine. It means that if there is the 1000 ml of urine, the kidney can concentrate about 1200 millimoles of metabolic waste. But normally, daily waste production in a healthy individual of 70 kgs is 600 millimoles of waste. So you can calculate it. If there is 600 millimoles of waste, so how much amount of urine the kidney must require to empty this? It's double amount. It's the double amount. For 1200 millimoles, we require the 1000 ml. For 600 millimole, we require 500 ml. It's the half amount of it. You can see here. We can half it and we can understand that minimum production of waste is 600, so we require 500 ml of water to clear it out. If the urine is less than 500 ml, so these products like urea, creatinine, and other metabolic waste accumulates in the body, and the urea and creatinine level begins to rise. It results in azotemia. Azotemia is different from uremia. Azotemia is initial stage and uremia is the advanced stage and it is symptomatic, but azotemia is asymptomatic. But this 500 ml of urine production is the minimal range for most of the individuals. But the individuals with hypercatabolic state, like they have severe traumas or burns or have high dietary protein intake, they have increased breakdown of proteins. So they have high amount of waste products, not 600 moles, but maybe they have 800 millimoles, maybe they can have 1000 millimoles, and it is normal for them. So their minimal urine output should exceed 500 ml to avoid waste accumulation in the body. So this was the cardiac output and the urine formation process, and it's the generalized and basic concept about how the urine is being formed in both kidneys under the influence under the time limit, and what is polyuria and oliguria. So this one was for today, and we'll discuss the glomerular filtration rate in series in the next videos. So uh, till then, goodbye, and I will see you in the next video.